I'll be talking about the signaling interplay between ULK1, which is the initiating autophagy kinase, and mammalian target rapamycin complex 1 signaling. And this is through Raptor phosphorylation on multiple sites, which hinders mTOR complex 1 signaling as well as substrate interaction of Raptor and downstream mTOR targets. So historically, it's been known that autophagy can cause inhibition of mTORC1 signaling, but the mechanism of this is unknown. So we wanted to determine whether or not ULK1 could directly modulate mTOR signaling, considering that ULK1 drives autophagy. When we overexpress kinase active ULK1 or wild-type ULK1, we can see a multi-site phosphorylation of Raptor, which is the scaffold protein of the mTORC1 complex. As we observe here with the Western blots, we see phosphorylation at 6 key sites, which is depicted in the cartoon picture of Raptor down below, which lies within the central region between the heat domains and the WD40 repeat domains. The kinase dead functions as a control in the Western blot, just to show that the kinase dead ULK1 was unable to induce multi-site phosphorylation. You'll also notice that there is a mobility shift as well of these phosphorylated Raptor protein. In order to test whether or not ULK1 was able to repress mTORC signaling as we originally proposed in our model, we overexpressed Rheb, which is a potent inducer of mTORC signaling, and we co-expressed XSK1 and immunoprecipitated this with and without ULK1 expression and with and without Rheb expression. As you can see in the second lane, when we overexpress Rheb, we can induce potent phosphorylation of SXK1 at the threonine 389 site, which is indicative of mTORC1 activation. And when we purified this and carried out SXK1 kinase assays against recombinant ribosomal protein S6 as a substrate, we can see increased SXK1 kinase activity. The interesting thing is that when we overexpress ULK1, we're able to completely block Rheb-induced mTOR activity, which is suggestive of a mechanism where Raptor phosphorylation by ULK1 can dominantly impair the actions of Rheb. So we considered the possibility that maybe ULK1 phosphorylation of Raptor might be blocking Raptor function to signal to downstream components. It's known that Raptor can bind to mTORC1 substrates through mTOR signaling motifs. A well-characterised one is 4-ABP1. To look at Raptor-substrate interactions, we utilised a far western approach, and this is depicted in this cartoon of 4-ABP1 resolved on gel, which is transferred to membrane and then cut into sections. When we incubate a lysate which contains Raptor, we'll see interaction of Raptor with 4-ABP1 on the membrane, and then we're able to build up just by regular primary and secondary antibodies for detection. So this is called a far western Raptor-Substrate Binding assay. As you can see with the western blots, or the far western blot, when we incubate wild-type Raptor unphosphorylated by ULK1, which is the second lane, we can see robust binding of MIC-tagged Raptor to the 4-ABP1. Of interest, when we use MIC-tagged Raptor that's been phosphorylated by ULK1, and you can observe this mobility shift, we robustly inhibit interaction of Raptor with 4-ABP1, and the kinase dead blocks some of this interaction. This is suggesting a twofold mechanism, where ULK1 by itself, even the kinase dead form, can block Raptor substrate binding, and it's known that Raptor and ULK1 are able to interact with each other. But ULK1 can further cause inhibition of mTORC1 signaling through phosphorylation of Raptor. And we see significance between the wild type and kinase dead ULK1. This is comparing lanes 3 and 4. So the mechanism we have, which adds to our current understanding of signalling interplays between mTOR and ULK1, is that under conditions when ULK1 is active, i.e. when energy is deprived in cells as well as amino acids, you have AMPK phosphorylation of ULK1. ULK1 becomes active but then autophosphorylates itself, which further activates ULK1, and ULK1 can turn off mTORC1 signalling through Raptor phosphorylation. And this will drive autophagy, which regenerates cells of new amino acids as well as energy, while repressing anabolic processes that mTORC1 drives, i.e. cell growth, through which would consume amino acids and energy. And this is the paper where most of this data is derived from. Thank you very much.